Hello everyone. Today we are discussing about the very important topic of the clinical research. It is a drug development process. In these slides, we are going to study the details about the drug discovery, different phases of the preclinical, phases of the clinical research, and the phase 4 study, FD approvals, a complete information about the drug development process. So let's discuss about the very first topic, very first step of the drug development process, and it is a drug discovery. Drug discovery is a first step in a drug development process in which of finding a new chemical compound that can help treat or manage a disease. Researchers discover drug by studying the disease process and design medicines to block or reverse them. Steps in a drug discovery, first of all, identification of drug candidate, synthesis and characterization, screening and testing for the therapeutic effect. If a compound shows good results, it moves to a drug development in clinical trials. The process is a long, costly, and complex. It takes 12 to 15 years for one drug to reach patient. Cost approximately about $900 million to $2 billion. Out of 5,000 to 10,000 compound tested, only one gets approved. Success require high expertise, scientists, researchers, project manager, advanced lab and technology, persistence plus luck. Ultimately, this process gives hope and relief to millions of patients because we have founded a new drug. It is going to help the millions of patients. Now, main stages of the drug discovery and development. First of all is the drug identification, drug validation, lead identification, lead optimization, product characterization, formulation and development, preclinical research, investigational new drug, clinical trial, phases 1, 3, new drug application, regulatory approval. Please see this detail about the drug development process. Drug discovery and development. Early drug discovery, preclinical study, clinical development, FDA review, post-marketing. In this target identification, target validation, hit discovery, we will discuss about the hit discovery detail, hit optimization, lead optimization, preclinical development. Phase 1, phase 2, phase 3, submission and approval. Phase 4. Discussed in a drug discovery, first of all, is the drug identification. Scientists first identify a biological target like protein, enzymes, or receptors. The target must play an important role in the disease. Example, in diabetes, the insulin receptor is the key target. This is a good example how how the target identification will take place in a drug discovery process. Target validation confirm that changing the target's activity can improve the disease outcome. Use laboratory experiments, genetics, and disease models. Steps in a drug discovery, hit identification. Screening thousands or millions of chemical compound to find hits. A hit is a compound that shows some activity against the target. Method. हिट का फुल फॉर्म है हाई थ्रूपुट स्क्रीनिंग। ये बहुत लार्ज रोबोटिक मशीन्स होती है जिसमें थाउजेंड्स और मिलियंस ऑफ मॉलिक्यूल को हम एक साथ फाइंड कर सकते हैं। रोबोट टेस्ट थाउजेंड्स ऑफ कंपाउंड्स क्विकली। कंप्यूटर एडेड ड्रग डिजाइन, वर्चुअल स्क्रीनिंग विद एआई एंड मॉलिक्यूलर मॉडलिंग। हिट टू लीड डेवलपमेंट एक बहुत इंटरेस्टिंग प्रोसेस है। हिट्स आर इम्प्रूव टू बिकम द लीड। Scientists add the variations of the chemical structure to improve the potency, strength of action, selectivity, acts only on the target, not others. So these are the lead. ये पिक्चर्स में आप देख सकते हैं हाउ थ्रूपुट हाई थ्रूपुट स्क्रीनिंग का जो स्टेप्स बताए हैं, किस तरह से थाउजेंड्स ऑफ इमेज क्रिएट हो रही है, किस तरह से ये रोबोटिक टेक्निक को यूज करके वन मॉलिक्यूल वन लीड मॉलिक्यूल डिसाइड करते हैं, फाइनल करते हैं। Steps in a drug discovery, lead optimization क्या है? फाइन ट्यूनिंग ऑफ द बेस लीड मॉलिक्यूल, एडजस्ट स्ट्रक्चर टू इम्प्रूव। जैसे कि आपने स्टडी किया होगा अबाउट द स्ट्रक्चरल एक्टिविटी रिलेशनशिप studied in the medicinal chemistry, different types of molecules are decided here. Efficacy, how well it works, bioavailability, how well it is absorbed in the body, reduced toxicity. At this stage, candidate drug are chosen for a preclinical testing. Timeline of the drug discovery, typically आप मान के चलिए कि it take 2 to 4 years before moving to preclinical stage. Out of 10,000 molecule screen, only one may become a marketed drug, or maybe sometime it also fails. So it's a very complex, very interesting, very time consuming and very costly process, जैसा कि आपने देखा है. प्रोडक्ट कैरेक्टराइजेशन. अब फाइनली जब लीड ऑप्टिमाइज हो चुका है, लीड आइडेंटिफाई हो चुका है, तो हम उसका प्रोडक्ट आइडेंटिफाई करेंगे, उसका डोजेज फॉर्म आइडेंटिफाई करेंगे, ताकि वो प्री क्लिनिकल स्टडीज से पहले एनिमल्स की बॉडी में पहुंचने से पहले हम उसका डोजेज फॉर्म डिसाइड करेंगे. तो फाइनली प्रोडक्ट कैरेक्टराइजेशन एंड फॉर्मुलेशन डेवलपमेंट विल टेक प्लेस. प्रोडक्ट का कैरेक्टराइजेशन होगा. When a new drug shows promise, it is studied for its size and shape, strength and weakness, toxicity, safety concern, biological activity, how it works in the body, mechanism of action, how it affects the disease. ये सारी चीजें प्रोडक्ट का कैरेक्टराइजेशन किया जाएगा. प्रोडक्ट के कैरेक्टराइजेशन होने के बाद उसका फॉर्मुलेशन एंड डेवलपमेंट होगा टू मेक द ड्रग इनटू अ सेफ, स्टेबल एंड इफेक्टिव टेबलेट, कैप्सूल एंड इंजेक्शन. Key pre-formulation studies: solubility, how well it dissolves in a different media, dissolution rate of drug release, stability, how long it lasts under the various conditions, solid state property, particle size, shape, and crystal forms. Finally, formulation and development activities: develop dosage form for a new chemical entities, improve the optimization existing formulation, develop controlled sustained release drugs, slow release, create a novel delivery system, example nano emulsion, self emulsifying system, colloidal system, ensure bioavailability, drug reaches bloodstream effectively. Now, finally, we are after the dosage form is decided. Now we come to the preclinical testing of the drug discovery and development process. Preclinical testing is done only on the animal studies before human trials. To evaluate the safety and efficacy of the drug before clinical trial, require a regulatory approval. जैसे कि आपने नाम सुना होगा, CPCSC गाइडलाइंस के हिसाब से गवर्नमेंट्स का अप्रूवल लिया जाता है कि हाउ मेनी एनिमल्स we are taking in the preclinical study, and everything required an approval, and it must follow the ICH guidelines, ethical and safe. Types of preclinical studies, जैसे कि pharmacological study in which the pharmacokinetic studies, absorption, distribution, metabolism and excretion, ADME, pharmacodynamics, how the drug affect the body, help decide dosage form, half-life and therapeutic effects. In preclinical study, toxicological studies are also performed, जैसे कि in vitro cell test check cell growth and effect, in vivo animal test check toxicity level, side effect, mode of action, correct animal species must be chosen since drug can be species specific. Key outcome: provide data on safety, metabolism and toxicity. Only drug proven safe and effective in animal get approval to move into the human clinical trial. After the preclinical study, we have to submit the investigational new drug application to the FDA. Before starting the clinical trial in human, drug developer must file an IND application with the FDA. The IND application includes the data from the preclinical and toxicity study, drug manufacturing details, quality, purity, consistency, clinical trial protocol, how studies will be conducted, any previous clinical research data, information about the investigators and developers, clinical research, clinical trials, studies in a voluntary people to test safety and efficacy of the drug, vaccines or a new treatment. Trials follow a study protocol designed by a researchers or manufacturers. Before starting, developers file an IND application, जैसा कि हमने study किया in previous slide. Researchers review existing data, then plan who can participate, what are the inclusions and exclusion criteria for the patient to participate in the clinical trial. Number of participants will be decided, study duration, dose and route of administration, parameter to assess, methods will be decided. Phase 0 microdosing study. Phase 0 is the first step in a clinical trial. First, in a human, the small studies, only 10 to 15 volunteers will be taken, very low Subtherapeutic doses will be provided to the patients. Purpose collect pharmacokinetics data for the absorption, distribution, metabolism, excretion studies. Help select the best drug candidate before larger trial. Phase 1, it is a safety and doses. First test in 20 to 80 volume tons, healthy or sometimes patients too. Purpose check the safety doses, range and side effect, studies in pharmacodynamics and mechanism of action, adjust dose based on animal study data. Around 70% of drug move to the phase 2. Now it's a phase 2 efficacy and side effect check. In this phase, it is conducted on 100 to 300 patients. Purpose to check the efficacy, does it work? Collect the more safety data for the drug, help determine the therapeutic dosage for the phase 3. About 33% of drug move to the next phase. Phase 3 is a confirming efficacy and safety, conducted on 300 to 3000 volume drugs. Purpose confirm the effectiveness, monitor adverse drug reaction, common and rare. In the phase 3, longer and larger studies detect the long-term side effect. Around 25 to 30% of drug progress further, data used to file for the marketing approval. After the phase 3, you can see here the phase 1, phase 2, phase 3, FDA review, phase 4. This is the complete process of the clinical trial phases. After the phase 3, new drug application will be filed, submitted to the FDA after the successful trial, tend to complete the story of the drug, preclinical to phase 3 data. It must include the clinical trial results, proposed labeling, how to use, safety update, drug abuse information, patent details. Purpose to provide drug is safe and effective for the public use. Thank you. In this, now the FDA review. The FDA review of the new drug application. And finally, it will take 6 to 10 months to review the new drug application. Complete new drug application reviewed for approval. If it is incomplete, it will be rejected. If drug is safe and effective, FDA works with the developers on labeling, prescribing information. FDA may ask for additional studies before approvals. Developers can appeal if they disagree with the FDA's decision. For phase 4 is a post-market surveillance conducted after the FDA approval. Purpose monitor long-term safety, efficacy, and cost effectiveness in a real-world use. Focus on pharmacovigilance, detecting and managing adverse drug reaction. So phase 4 studies are always conducted only after the FDA approval. If FDA approval is not conducted, then the phase 4 study is not going to happen. And in the post-marketing surveillance, thousands and thousands of patients are studied. It may lead to the labeling changes sometimes, risk management plans, safety warns, dosage change, ongoing studies ensure the drug safety throughout its market life. In this complete drug development process, we have covered about the details of the preclinical studies, the drug discovery process, and the clinical trials, the details about the drug development process. I hope you understood the detail about the drug development process. Thank you for the listening, for your time. We are so thankful that you have given us the time. Thank you for the listening. Here we have studied